Biology, the science study of life. Environment interacting with animals. Genetic makeup influences the development of behaviors. While experimental demonstrations of genetic influences on behaviors accumulate, research also reveals that environmental conditions modify many of the same behaviors. Environmental factors such as the quality of the diet, the nature of social interactions, and opportunities for learning can influence the development of behaviors in every group of animals. Dietary influence on mate choice behavior. One example of environmental influence on behavior is the role of diet mate selection by Drosophila majvensis, which mates and lays its eggs on the rooting tissues of Coctos de Mujavensis populations in Baja California, Mexico, breed almost entirely on Agria cactus, whereas most populations in Sonora, Mexico, and in Arizona were use organ by cactus. These cacti serve as food for the developing larvae. When the Majovenses from Baja and Sonora were raised on an artificial banana based medium in the laboratory, researchers noticed that females from the Sonoran population tended to avoid mating with Baja males. Following up on these initial observations, Williams, Etish, and Michel of the University of Arkansas showed that the food eaten by the Majovensis larvae strongly influences later mate selection by females, especially those from Sonoran populations, as shown in this video. But why do Sonoran females avoid Baja males that develop on some diets, but not those that develop on other diets? In other words, that is the approximate cause of that behavior. Marco of Arizona State University and Eric Tolson of the University of New Mexico proposed that the physiological basis of the observed mate preference was differences in hydrocarbons in the exoskeleton of the flies, as shown in this video. And notice that this was a responsible hypothesis since, in addition to using courtship songs and other sensory information, Drosophila used their sense of taste to assess the hydrocarbons in the exoskeleton of possible mates. To test the influence of exoskeleton hydrocarbons, Eaters and Hernes raised Baja males to an artificial medium and then performed them with hydrocarbons excreted from the exoskeletons of males from the Sonoran population. Instead of rejecting these Baja males, Sonoran females accepted them as frequently as they accepted Sonoran males. Ethers and Aronson's study showed how the effects of nutritional environment can modify critical behavior. Next, we will see, as shown here, the social environment and aggressive behavior. The discoveries of genetic influence on mating behaviors in males prairie voles are complemented by parallel studies of another monogamous rodent. The California mouse, Brimoicos California, like prairie voles, male California mice are highly aggressive toward other mice and provide extensive parental care. In contrast to prairie voles, however, even male California mice that have not mated are already aggressive. Janet Bester Midrith and Catherine Merler of University of Wisconsin at Madison studied what influence the social environment might have on the behavior of male California mice. To manipulate the early social environment, they placed newborn California mice in the nests of white-footed mice, a species in which males are not monogamous and engage in little parental care. They also placed newborn white-footed mice in the nests of California mice. This cross-fostering, in which the young of each species were placed in the nests of the other species, or altered altered the behavior of both species, as shown this diagram. For instance, when a male California mice raised by white-footed mice crawled away from the nest, they were not retrieved as frequently as those raised by their own species. When these cross-fostered California mice became a parent, they too spent less time retrieving their pups. The cross-fostered California mice were also less aggressive toward intruders. In contrast, male white-footed mice raised by California mice were more aggressive than those raised by white-footed mice. In one study by Mr. Midrith and Miller, the brains of California of mice in one study of California mice raised by white-footed mice were found to contain reduced levels of the same neurotransmitter AVP associated with elevated aggression and parental care in male prairie voles. The fact that cross-fostered California and white-footed mice adopted some of the behaviors of their foster parents suggests that experience during development can lead to changes in parental and aggressive behaviors in these rodents that can be passed from one generation to the next. Learning. One of the most powerful ways that environmental conditions can influence behavior is through learning, the modification of behavior based on specific experiences. Learned behaviors range from the very simple, such as imprinting, which results in a young bird following a particular individual it has learned to recognize as its parent, to the highly complex. Before we consider some complex forms of learning, let's look briefly at another very simple habituation, as shown in this video. Habituation is a loss responsiveness to stimuli that convey little or no information. Examples that are widespread. Hydra contracts when disrupted by a slight touch of its repeatedly disrupted by such a stimulus without any further consequences. It stops responding. Many mammals and bears recognize alarm calls of members of their species, but they eventually stop responding if these calls are not followed by an actual attack. The cry wolf effect. In terms of ultimate causation, habituation may increase fitness by allowing an animal's nervous system to focus on stimuli that signal the presence of food, mates, or real danger instead of wasting time or energy on a vast number of other stimuli that are irrelevant to the animal's survival and reproduction. Next. Let's shift our attention to some more complex form of the learning, spatial learning. Every natural environment shows some degree of spatial variation. For instance, sites suitable for nesting may be more common of an organism in some places than in others. As a consequence, the fitness of an organism may be enhanced by the capacity for spatial learning, which is the modification of behavior based on experience with the spatial structure of the environment, including the locations of nest size, hazards, food, and prospective mates. In a classic experiment done in 1932, Nico Tinbergen studied how digger wisps find their nest entrances. Tinbergen moved a circle of pine cones that had previously surrounded a nest entrance and observed that a wasp landed in the center of the West intense and observed that the wasp landed in the center of the pine cones, even though the nest entrance was no longer there, as shown in this video. The wisp was using the pine cones as a landmark or location indicator. The use of landmarks is a more complex cognitive mechanism than a taxis or a kinesis. More since it involves learning, the wisp flies toward a stimulus, in this case, the center of the pine cones, as in a taxis, but the stimulus is an a pituitary, as shown here. A pituitary landmark, the animal must learn rather than a constant stimulus such as light. One nest entrance may have pine cones around it, while another may be next to a pile of stones. Each wisp has to learn the unique landmarks of each individual nest site. 10 Bergen's experiment reveals that for spatial learning to be a reliable reliable way to navigate through the environment, the landmarks used must be stable within the time frame of a particular activity. For example, the pine cone that indicate the presence of the wisp's nest should have allow a probability of moving. Using unreliable information for learning may result in a significant cost to the animal. If a nesting wasp learns to locate its nest using objects that might be low away, for instance, the cost may be an inability to locate and provision the nest, and consequently reduce the reproductive success of the wasp. Because some environments are more stable than others, animals may use different kinds of information for spatial learning in different even environments. Lucy Alding Simi and Victoria of the University of Edinburgh hypothesized that three-spined stickleback fish from stable pond environments would rely more on landmarks than would sticklebacks from um, or from more changeable river environments. Alding, Simi, and Breath Breather um, weight trained 20 sticklebacks from rivers and 20 from bands to navigate a T-shaped maze to reach a reward, a combination of food and other fish and sticklebacks could school with. In the first phase of the experiment, the researchers placed the reward at one end of the maze and marked the correct direction to turn for the reward with landmarks in the form of two plastic plants. First, that learning to find the reward were given a second set of trials in which Alding, Simi, and Braithwaite put toward at both ends of the maze and switched the location of the landmarks. The results showed that the sticklebacks from stable bound environments located the reward using combination of learning which direction to turn and landmarks, while massive fish learned simply to turn in a particular direction. These results suggest that the degree of environmental variability influences the spatial learning strategies of animals.